everyone, and welcome to the Respiratory System Part 5. In this section we'll be looking at internal and external respiration processes, the binding of oxygen and haemoglobin, and also regulation of respiration. So as we know, gas exchange occurs between the blood and the air at the alveoli of the lungs. Oxygen diffuses into the blood and carbon dioxide diffuses out. If you remember from our previous slide that each alveoli is surrounded by a cobweb network of capillaries, and this dramatically increases the surface area available for gas exchange. So when we consider the proximity of the capillary next to the alveoli, we can see that blood with red blood cells and haemoglobin molecules travel along the capillaries and bind to the oxygen. Carbon dioxide will transfer from the capillaries and into the alveoli. At a molecular level, we can see that the process involves Hb, or haemoglobin, binding with the molecule of O2, and this unloads into the tissue cells. The concurrent process is carbon dioxide binding with water in the systemic capillaries. This forms carbonic acid, which is transported in the blood as a hydrogen ion and a bicarbonate ion. Internal respiration is the exchange of gases between the blood capillaries and the cells. The level of O2 within the blood will be between 100 to 105 millimeters of mercury, whereas in the cells it's approximately 40 millimeters of mercury. At rest, approximately a quarter of the available oxygen will enter the cells, and this will obviously increase with increased metabolic demands, for example during exercise. 1.5% of oxygen is physically dissolved in the blood plasma. This means that approximately 98.5% is chemically bound to haemoglobin molecules. Haemoglobin is capable of unloading carbon dioxide and absorbing oxygen 60 times faster than plasma. Haemoglobin consists of a protein called globin and a pigment called heme. The heme molecule contains 4 atoms of iron, each of which can combine with a molecule of oxygen, and they bind in an easily reversible reaction to form oxyhemoglobin. The partial pressure of oxygen in the blood plasma is the most important factor in determining the extent to which O2 combines with haemoglobin. Venous blood entering the lungs has a partial pressure of 40 millimeters of mercury and a haemoglobin saturation of 75%. Oxygen will diffuse from the alveoli because of the higher partial pressure of oxygen, approximately 100 millimeters of mercury, into the plasma, and this follows with our laws of physics from last session, where the substance will want to move from areas of a high concentration or pressure to areas of a lower concentration or pressure. This diagram is available in your textbooks, and it's not essential to know all the figures on this. What I do want you to be aware of is that at lower oxygenation levels, haemoglobin shows a much higher affinity for saturation of oxygen. So if there's less oxygen around, haemoglobin will more readily bind with what is available. Whereas we can see that this graph tapers off at the top and forms a plateau. At higher oxygen pressures, haemoglobin shows a lower affinity for binding with oxygen. Diffusion will raise blood plasma pressures of oxygen. This causes an increased uptake of oxygen by the haemoglobin. In tissue capillaries, this process is reversed. As blood enters the capillaries, the plasma pressure of oxygen is greater than the pressure of oxygen in the fluid surrounding the capillaries. This results in diffusion across the capillary membranes to the cells of the tissues. This diagram, again from your textbooks, shows the transport of carbon dioxide and oxygen within the blood between the alveoli and the systemic capillaries. Factors which will decrease haemoglobin binding include acidity. This is known as the Bohr effect, and as acidity increases, i.e. the pH decreases, the affinity of hemoglobin for oxygen decreases. This is because hydrogen ions will react with hemoglobin, causing oxygen to be released. This creates a buildup of either lactic acid or carbonic acid. Carbon monoxide concentration will decrease hemoglobin binding, and BPG, a substance found in the blood, will also bind to hemoglobin, lowering its affinity for oxygen. At high altitude levels, there will be an increased level of BPG. This will result in more delivery of oxygen to the tissues under a decreased partial pressure of oxygen. This is referred to as acclimatization. Other factors that can affect blood oxygenation include depressed respiratory drive, for example paralysis of the respiratory muscles, increased resistance in the airways, decreased compliance, or chest wall abnormalities, decreased surface area for exchange, or increased thickness of the membranes and reduced ability of the blood to transport oxygen. Increased body temperature will result in increased blood oxygenation. Certain medications and psychological states will also have an impact on blood oxygenation. As the body ages, alteration in lung and chest wall compliance may result in increased thoracic rigidity, decreased elastic recoil of the lungs, enlarged alveolar ducts and sacs, fewer alveoli, and a smaller alveolar surface available for gas exchange. This will ultimately result in decreased oxygenation levels. As a result, lung function decreases with age. Respiration is regulated by the respiratory centre within the brainstem. The key areas are the pneumotaxic area and the apneustic area, and the medullary rhythmicity centre, which includes an inspiratory and an expiratory area. Inspiration and expiration, as we know, is produced by contraction and relaxation of skeletal muscles, along with pressure gradients in the air. The movement of the skeletal muscle is a response to activity in somatic motor neurons in the spinal cord, and the activity of these neurons is controlled by descending tracts from neurons in respiratory control centres in the medulla oblongata and neurons within the cerebral cortex. Motor neurons stimulate respiratory muscles and are controlled by two major descending pathways, one that controls voluntary breathing and another that controls involuntary breathing. Unconscious, rhythmic control of breathing is influenced by sensory feedback from receptors that are sensitive to partial pressures of carbon dioxide, oxygen, and acidity levels of arterial blood. So the medullary rhythmicity centre includes the inspiratory area and the expiratory area. If we look at normal quiet breathing, we can see that the inspiratory area is active for 2 seconds and then inactive for 3 seconds. During activity, the diaphragm actively contracts and there is normal quiet inspiration. During inactivity, the diaphragm relaxes, followed by elastic recoil of the chest wall and lungs, resulting in normal quiet expiration. However, during laboured breathing, the inspiratory area will activate the expiratory area, as well as recruitment of the diaphragm and the external intercostal muscles. Other lesser muscles are recruited in order to create a forceful inspiration. The expiratory area will recruit the internal intercostal and abdominal muscles, causing forceful expiration. The pneumotaxic area transmits inhibitory impulses to the inspiratory area within the medulla, and the apneustic area transmits stimulatory impulses to the inspiratory area when the pneumotaxic area is not active. So the apneustic area prolongs inspiration and inhibits expiration. This diagram from your textbooks shows the regulation of respiration by the central nervous system. We can see at the top the cerebral cortex will control voluntary breathing by sending signals down the spinal cord via motor neurons to the respiratory muscles. Automatic breathing is a result of the influence of the apneustic and pneumotaxic area on the medulla oblongata, and feedback is provided by aortic and carotid bodies, as well as chemoreceptors in the medulla oblongata. So from the previous diagram, we know that cortical influences are responsible for the voluntary control of breathing. There is an inflation reflex, which is controlled by stretch receptors within the bronchioles. Impulses will be sent along the vagus nerve to the inspiratory area and the apneustic area, causing inhibition of inspiration. This is known as the Herring-Brewer reflex and prevents overinflation and distension of the lungs. During this module, we talk about homeostasis and maintaining balance within the body. So we'll imagine that some stimulus disrupts homeostasis and causes an increase in the arterial partial pressure of carbon dioxide or a decrease in oxygen. Receptors within the medulla and the aortic and carotid bodies send nerve impulses to the inspiratory area within the medulla oblongata. Nerve impulses are sent to effectors in the diaphragm and other muscles of respiration, causing these to contract more forcefully and frequently, thus increasing ventilation. There is therefore a decrease in the arterial level of carbon dioxide and an increase in acidity and oxygenation. When this response brings the levels back to normal, this feedback loop switches off the process. So we've seen that levels of oxygen, carbon dioxide and hydrogen ions within the blood are all responsible for the regulation of respiratory centre activity. Carbon dioxide and hydrogen ions are the most important. Carbon dioxide is dissolved within the cerebrospinal fluid, and this fluid also bathes receptors sensitive to hydrogen ions within the medulla. Stimulation of these receptors will result in a 70% increase in the rate and depth of respiration in response to increased carbon dioxide levels. The carotid and aortic bodies are responsible for the other 30% of response to raised carbon dioxide levels. They also increase ventilation in response to a rise in hydrogen ions or a large drop in oxygen levels. The chemical and nervous means of regulating the activity of the respiratory centres act together to adjust the rate and depth of breathing. Obviously, exercise will increase the requirement for oxygen and produce more carbon dioxide. Increased ventilation is therefore necessary to remove extra carbon dioxide and supply the tissues with extra oxygen. Alveolar oxygen and carbon dioxide levels will remain constant. This slide links back to our previous sessions on all areas within the respiratory tract and how they respond to a variety of sensory stimuli. Pungent odours will irritate nerve endings in a nasal mucosa, and as a result there will be short inspirations with forced expirations, and the glottis will open in sneezing. Should a bolus of food contact the pharynx, there will be inhibition of inspiration during swallowing. Irritant contracting the larynx or the trachea will again result in short inspiration with forced expirations and the glottis closed. The glottis will open suddenly to blast the air and carry the irritant out, and this is the coughing reflex. Pain, heat, cold stimuli to the nerve endings in the skin will result in sharp inspiration after pain or cold, or an increased rate and depth of breathing with heat. That concludes our sessions on the respiratory system.